Good morning. It's October 25th, and this is your daily brief in science. Here's everything you need to know. Recent research highlights the pressing need for sustainable alternatives to traditional mineral fertilizers, which can have detrimental effects on ecosystems. The findings provide critical insights into the use of biochar as a means to enhance agricultural practices and improve soil health. In regions with sufficient water supply, a mixed biochar with a pH range of 6 to 8, applied at rates between 41 to 80 tons per hectare, has been identified as optimal. In contrast, in water-scarce areas where soil pH exceeds 8.5, biochar with a pH of 6 or lower, applied at rates greater than 80 tons per hectare, proves to be more beneficial. The study also revealed that combining biochar with phosphogypsum significantly reduces cadmium levels in both soil and edible parts of Chinese cabbage, while simultaneously increasing the fresh weight of the crop. This research underscores the importance of microbial interactions and organic amendments in sustainable agriculture, particularly in alleviating salinity's adverse effects on crop yields. Moreover, the potential of novel recycled fertilizers, such as bio-based nitrogen and phosphorus fertilizers, is emphasized as a way to enhance nutrient cycles in sustainable farming. Organic amendments have been shown to effectively improve soil conditions, leading to better nutrient availability and enhanced vegetation growth. Phosphorus is crucial for plant growth but is often limited in soils, especially in acidic conditions where it binds to minerals, reducing its accessibility. Field trials indicate that biological treatments at reduced fertilizer doses can improve both crop yield and quality, while boosting microbial activity in the soil. Returning organic materials to fields is vital for enhancing soil carbon sequestration and promoting agricultural sustainability. The study calls for additional research to fully leverage the potential of microorganisms in achieving food security and fostering environmental health. Recent research has unveiled that polycyclic aromatic hydrocarbons, or PAHs, such as pyrene, may account for 10 to 25% of the carbon present in space, highlighting their significance in the chemistry of interstellar clouds. Utilizing the Green Bank telescope, researchers have successfully detected cyanopyrene, a modified version of pyrene, in the Taurus molecular cloud, where it comprises about 0.1% of the carbon content. This discovery establishes pyrene as the largest PAH identified in space, consisting of 26 atoms, and contradicts earlier beliefs regarding its ability to endure the extreme conditions of star formation. Notably, pyrene was discovered in an environment with a temperature of only 10 Kelvin, prompting questions about how PAHs can form under such frigid conditions. The findings contribute to a deeper understanding of the chemical processes that occur before planet formation and indicate that essential life-building molecules may be more widespread in space than previously recognized. This research bolsters the hypothesis that complex organic molecules necessary for life could withstand the conditions leading to star and solar system formation. The implications of this research suggest that the conditions required for life to emerge on Earth may not be unique, but rather a common phenomenon throughout the universe. Astrochemist Ryan Fortenberry has pointed out the importance of glyceric acid as a transitional molecule that bridges prebiotic chemistry and biochemistry, emphasizing its role in the origins of life. Understanding how molecules like glyceric acid form in space is crucial for unraveling the mysteries surrounding life's origins on Earth, as highlighted by researcher Ralph Kaiser. This study, published in the journal Science, was led by Gabby Wenzel, with Brett McGuire and Ilse Cook serving as senior authors, showcasing a collaborative effort in advancing our understanding of space chemistry. The research received funding from various organizations, including NASA and the National Science Foundation, underscoring the collaborative nature of this scientific endeavor. A recent perspective paper published in the journal Joule introduces a method called electro-agriculture, which aims to replace traditional photosynthesis with a solar-powered chemical reaction. This innovative approach utilizes solar panels to convert carbon dioxide and water into acetate, an organic molecule that genetically engineered plants can use in a hydroponic system. Lead researcher Robert Jinkerson emphasizes the potential for growing food in controlled indoor environments, effectively decoupling agriculture from reliance on natural sunlight. This method could drastically reduce agricultural land use in the U.S. by up to 94%, paving the way for food production in urban centers, arid regions, and even outer space. However, despite its promise, electro-agriculture faces significant challenges and requires further research and development before it can be commercialized. The research team has developed a tandem electrolysis process that achieves nearly 90% selectivity for acetate, significantly enhancing production efficiency. Current efforts are focused on engineering plants that can utilize acetate alongside photosynthesis, with the goal of creating plants that rely solely on acetate for energy. Researchers are working on reactivating a dormant metabolic pathway in plants that typically remains inactive after germination. Preliminary experiments have shown success with various crops, including lettuce, rice, and tomatoes, indicating the method's potential for diverse agricultural applications. The urgency for innovative agricultural solutions is highlighted by the fact that around 14 million households in the U.S. faced food insecurity in 2023. Unlike conventional farming, electro-agriculture requires electricity, which can be sourced from renewable or non-renewable sources. Overall, electro-agriculture achieves about 4% efficiency, which is 4 times higher than traditional photosynthesis, promising a significant advancement in food production. Recent research has brought attention to MEF2C haploinsufficiency syndrome, or MHS, a severe form of autism associated with mutations in the MEF2C gene. The study highlights how a specific genetic mutation disrupts the balance of brain cells during development, contributing to autism. Findings indicate that MHS organoids show an excess of glial cells and a deficiency of inhibitory neurons, resulting in electrical signaling imbalances typical of autism spectrum disorder. Researchers discovered that by adding specific microRNAs, or miRNAs, to patient-derived organoids, they could restore a more typical balance of neurons and glial cells. This research identified nearly 200 genes regulated by the MEF2C gene, with 3 particular miRNAs playing a crucial role in brain cell development. Additionally, an experimental drug named nitrosynapsin has shown promise in partially correcting neuron imbalances in organoids, suggesting potential new treatments for autism. However, further research is necessary to evaluate nitrosynapsin's effectiveness in addressing symptoms of MHS and other forms of autism. In a related study, researchers are investigating the use of lipid nanoparticles to deliver Cas9 mRNA for gene editing in utero, which aims to correct genetic conditions before symptoms manifest. This innovative approach could potentially prevent genetic neurodevelopmental conditions like Angelman syndrome and Rett syndrome by addressing anomalies during critical brain development phases. The lipid nanoparticle tool has successfully transfected a significant portion of brain stem cells in mouse models, demonstrating substantial gene editing potential. This research, led by Aijun Wang and published in ACS Nano, showcases the collaborative efforts between UC Davis and UC Berkeley, and it received funding from various organizations, including the National Institutes of Health, underscoring its importance in the field of neurodevelopmental research. A recent study published on October 23, 2024 in the BMJ has revealed significant patterns in global suicide rates, analyzing data from over 1.7 million cases across 740 locations in 26 countries from 1971 to 2019. The research found that suicide risk peaks notably on Mondays and New Year's Day, with approximately 15 to 18% of total suicides occurring on Mondays. New Year's Day consistently showed increased suicide rates across all countries studied, particularly among men, while Christmas Day exhibited mixed results depending on the region. Weekend suicide rates varied significantly by region, generally lowest on Saturdays and Sundays in North America, Asia, and Europe, but higher in parts of South and Central America, Finland, and South Africa. The study identified South Korea, Japan, South Africa, and Estonia as having the highest suicide rates, while the lowest rates were recorded in the Philippines, Brazil, Mexico, and Paraguay. Men and individuals aged 0 to 64 years showed higher suicide rates compared to women and those aged 65 and older. Researchers suggest the increased risk on Mondays may be linked to the broken promise effect theory, where unmet expectations lead to negative moods, compounded by heightened work pressure. These findings are crucial for developing targeted suicide prevention strategies and alert policies, particularly focused on specific days of the week and holiday periods. Further research is recommended to explore potential causes of these trends, including workplace pressure and alcohol consumption during holidays. The study also acknowledges limitations, such as potential underreporting and misclassification of suicide data in certain regions. Individuals in crisis or experiencing emotional distress are encouraged to reach out to the Suicide and Crisis Lifeline at 988. This has been your daily brief in science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. We'll be back Monday with everything you need to know.